Hello everyone, welcome back to Perfect Lab. In today's video, we will be learning about the SIM test, which is an important biochemical test used in microbiology to identify bacteria based on three characteristics: hydrogen sulfide production, indole production, and motility. In this video, I will also explain the principle of the test, the medium composition, and demonstrate the procedure step by step and show you how to interpret the result accurately. If you are new to the channel, please like, subscribe, and turn on the notification bell for more microbial practical tutorials. So let's get started. The sulfide indole motility test, or in short SIM test, is a biochemical test used to determine three important characteristics of bacteria in a single medium. 1. Hydrogen sulfide production. 2. Indole production. And 3. Motility. The SIM test is commonly used to identify and differentiate members of the Enterobacteriaceae and other gram-negative bacteria. The SIM test is based on the ability of bacteria to produce hydrogen sulfide from sulfur-containing compounds, produce indole from the amino acid tryptophan, and move through a semi-solid medium using flagella. So let's look at the hydrogen sulfide production. Bacteria that produce the enzyme thiosulfate reductase reduce sodium thiosulfate in the medium to hydrogen sulfide. The hydrogen sulfide then reacts with the ferrous ammonium sulfate in the medium to form a black precipitate, which is also known as ferrous sulfide. For the indole production, bacteria that have the ability to produce the enzyme tryptophanase break down tryptophan into indole, pyruvate, and ammonia. And after the addition of the Kovac reagent, the indole reacts with the reagent to produce a red ring at the surface of the medium as a positive test. And for the motility, motile bacteria migrates away from the start line through a semi-solid agar, causing diffuse growth. So for a motile or a positive result, growth diffuse away from the start line as shown over here. And for a non-motile bacterium or a negative result, growth occurs along the start line. So for a positive hydrogen sulfide result, there is a black precipitate in the medium as shown over here. And for the positive indole result, a red ring at the surface after adding the Kovac reagent is seen. And for a motile bacterium, there is a diffuse growth away from the start line, and for a non-motile bacterium, growth only occurs along the start line as shown in these two pictures. So in summary, we can say that the hydrogen sulfide production is shown by a black precipitate formation in the medium. The indole production, a positive result, is shown as a ring red at the surface after adding the Kovac reagent. And for the motility, for a motile bacterium, growth diffuse away from the start line, and from a non-motile bacterium, growth occurs along the start line. Composition of SIM medium and their importance. The SIM medium contains peptone, sodium thiosulfate, ferrous ammonium sulfate, and approximately 0.35% of agar. Let's look at the first component, which is peptone, and which is rich in tryptophan. So peptone provides nutrients for growth and supplies the amino acid tryptophan, which is essential for the indole production. The second is sodium thiosulfate, which serves as a sulfur source or a substrate for bacteria that produce hydrogen sulfide. Third, ferrous ammonium sulfate, which acts as an indicator for hydrogen sulfide production. And fifth, approximately 0.35% agar to create a semi-solid medium that allows detection of bacterial motility. So let's look at the same medium test in a tube. So for Indole production, after stabbing and incubating and adding our Kovac reagent, Indole produces form a red ring at the surface as shown over here. For hydrogen sulfide production, hydrogen sulfide producers form a black precipitate along the stop line, which is usually in the bath as shown here. And for motility, motile bacteria growth diffuse away from the stop line, and for a non-motile bacteria, growth is only seen only along the stop line. So in summary, each component of the same medium plays a specific role in detecting hydrogen sulfide production, Indole production, and motility to allow differentiation of bacteria based on these three characteristics. Let's look at the step-by-step procedure for performing the same test. The first is to label the same medium tube with the sample identification, date, and other necessary information. 2 is to flame to sterilize the inoculating needle until it's red hot and then allowed to cool. 3 is using aseptic techniques. Pick a well-isolated colony from a fresh bacterium culture as shown here. And 4, stab the same medium straight down through the center to about 2/3 to 3/4 of the depth, and then withdraw the needle along the same path without striking the surface. And 5th, incubate at 35 to 37 degrees Celsius for 18 to 24 hours. And the last step, after incubation, add 3 to 5 drops of Kovac reagent to the surface of the medium and then observe for the development of a colored ring within 1 to 2 minutes. So some important notes. The first is that use a pure and a fresh, which is 18 to 24 hour culture for accurate results. To incubate for 18 to 24 hours. And do not read result too early or too late. And always follow aseptic techniques throughout the procedure to avoid contamination. So in summary, the step-by-step procedure for performing the same test involves 1, to label the tube, 2, sterilize the inoculating needle and allow it to cool, 3, pick a pure colony, 4, inoculate the medium, 5, incubate, and 6, add your Kovac reagent. Interpretation of same test result. So 1, for hydrogen sulfide production, bacteria that produce hydrogen sulfide react with the ferrous ammonium sulfate in the medium to form a black precipitate, which is also known as ferrous sulfide, to indicate a positive result, whereas no black precipitate formation indicates or shows a negative result. For Indole, bacteria that produce Indole from tryptophan will react with a Kovac reagent to produce a cherry red ring at the surface of the medium as shown here to give a positive result, whereas yellow or no color change on the surface indicates a negative result. For the motility, motile bacteria migrates away as shown here from the stop line through the semi-solid agar, causing diffuse growth to indicate a positive result, whereas growth confined to the stop lines indicates a non-motile bacteria or a negative result. quality control. So in running these tests, it is best to use known bacteria that shows a positive result and a negative result for each of the parameters. So for hydrogen sulfide production, Proteus mirabilis gives a positive result, whereas E. coli gives a negative result. For Indole production or Indole formation, E. coli gives a positive result, whereas Klebsiella pneumonia gives a negative result. And for the motility test, E. coli gives a positive result, whereas Klebsiella pneumonia gives a negative result. Limitation or some important tips to know. 1. Heavy blackening may obscure the observation of motility as shown in the pictures here. And also, Kovac reagent must be added after incubation, because adding it before incubation invalidates the Indole test. And a pure culture is essential for accurate results. Some common organisms and their expected result in the same test. So I'll leave this part for you to go through. Now let's look at the clinical importance of the same test. So the same test is useful for 1. Differentiating members of the Enterobacteriaceae. 2. Detecting hydrogen sulfide producing bacteria such as Proteus and Salmonella. 3. Identify Indole-positive organisms such as E. coli. 4. To distinguish motile bacteria from non-motile bacteria. So thanks for watching, and if you found this video helpful, please like, share, and subscribe to Perfect Lab. Leave your comments or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.